Welcome to the lab technique series. So microbes are present on all lifeless surfaces, so inanimate surfaces. So that means that there are many possible sources of contamination in the lab. And a microbiologist dreads the big C, contamination. Sometimes, despite one's best effort, contamination is unavoidable. But there's a skill or technique that, when performed properly, significantly reduces the chance of contamination. So the first skill I think that all baby scientists, baby microbiologists will learn is that of aseptic technique. So aseptic technique, as the name suggests, it's a technique and it involves a systematic transfer of microbial sample, whether it's fungus, bacteria, etc., from one source. So in microbiology, the source tends to be broth or the soup, so liquid media, or agar, the solid media, on plates. So from one source to another without introducing any contaminants. So contaminants would be the microbe that you do not want to grow. You just want your sample of interest to grow. And so you want to do that by sterilizing the tools in between each transfer. So what I'm going to do now is the process of inoculating the plates. This time I'm using spore solutions versus agar plug. So I'll take a known volume, say 100 microliters or 0.1 ml, I think. What's the math? And then I'm just going to put it into the center of the Petri dish that I'll show you, it's a square plate, and let it dry or evaporate. Yeah, let it dry, and then I put the cover on, wrap it up with paraffin to seal it, and then incubate it under the light humidity and temperature conditions that the fungus grows best under. So yeah, it's simple. It's just one way to let your fungus grow. And if it's bacteria, I would use the required media, and it would be kind of the same process. You put the known volume on the particular media, and you could either spread it with a glass rod / hockey stick, but I'm not doing a spread plate. I'm just going to do a point inoculation. So that's what you'll see me do. Before I go ahead and actually put the spores of the fungus onto the agar plate, I like to do a quality control. So I like to check to ensure that the sample that I'm taking is free of contamination. So just only my fungus of interest is in the water that contains the spores, and the shape is what it should be. So I'm just cleaning the hemocytometer, so the counting chamber, the thing I use to count, ensure that I have a standard, like a high enough amount, so maybe like 1,000,000 spores. And the counting chamber was created originally for counting blood cells, so it's called a hemocytometer. That's the vial with the spores, and I'm just going to quickly flame them out once I put the tip onto my pipette. And I accidentally knocked over the pipette container, so I have to manually put them all back into the box. It's a good thing that I'm working in a sterile environment, so it's fine. But once I'm done with that, I will then take 10 microliters, so not a lot, of the spores in water and put it onto each side of the counting chamber. So in total, it will be 20 microliters. I'll then put on a glass slide cover slip, take it to the microscope, and use my spore counting template. So different lab will have their own unique way of counting the spores, but it's relatively pretty much the same, but in terms of the rules that they'll use. And once that's done, then I'll go ahead and actually start the process of inoculating the plate, which will be the start of the experiment. What I'm going to do is, before I inoculate the plate with the fungus, so that it initiate the inoculum, I'm just going to run a quality control check on the spores that were in the little white tube to ensure that what I'm going to put on the plates are what I want. So I'm going to turn the light on. It's there. See, it comes on, and I'm going to set up I'm going to look through here. See, it's blurry. So I'm going to put it into focus and then count the spores in the slide. So I have my spore count template here, and I'm going to count how many spores I see in the different squares that I selected. I'm just going to spit out a number, and that will give me the final concentration. So it's at my starting concentration. Those little dots are spores of a fungus. And those squares that you're seeing, those are the counting chamber. I'll link a video to the hemocytometer counting that I did. But yeah. I retrieved the pre-made agar plates from the 4 degrees Celsius fridge, and as a standard practice, I avoid making the plates on the same day I inoculate them, but if it's unavoidable, I make them up first thing in the morning, then inoculate in the afternoon. So inoculation is a process by which the inoculum, a pure culture of the microbe, in this case, the spore suspension, is transferred to the growth medium, which is the agar plate, using techniques that minimize the introduction of unwanted microbes, aka contaminants. And labeling is a crucial practice in the lab. I label with my initials, the date, the project code, and the pathogen ID. Since the fungal sample passed the quality control test, I'll take 100 microliters or 0.1 ml aseptically from the vial and pipette it into the center of the agar plate. I'll repeat this process for however how many plates that I'll be using in the next phase. So I think I have about 8. Once I'm done, I'll stack the plates, cover off, allowing them to dry so that the water that the spore is in will evaporate, leaving just the spores on the agar plate. And the agar plate contains all the nutrients that this particular pathogen needs to grow. See, it looks like there's nothing on the plate. Looks like there's nothing on the plate. I'll replace the lids once they've dried and seal them with paraffin, which is that stretchy material. I'll spray everything back down with ethanol before I leave. Now I have to go next door to put these in the incubator. I don't have the light dark capacity on this one, so I'm going to incubate them in the growth chamber. And that's all for my Monday. I'm going to take these to get them incubated so that they can grow. So each type of microbe has a specific growth requirement. Example, specific nutrient type, whether they want lots of vegetable, lots of sugar, different substrate type, different environmental conditions, example, pH, light, and temperature. As I said, the incubator that I usually use is under, not construction, they're replacing the light, I think. So I'm going to put them in the growth chamber, and I use a standard light dark cycle, standard temperature, normal humidity. Come. Uh-oh. 2 hands. So because the fuse is out in the incubator that I usually use, I'm going to put them here in the growth chamber, the same conditions, and I'll collect them next week. Okay, bye.